Good morning everyone. Today topic is biomolecule chapter from NEET syllabus. In general, whenever students study this biomolecule chapter, they feel little difficult of these structures of amino acids. As the matter of the fact, when we compare the structures one amino acid to another, it is very easy to keep remember. Total amino acids are 20. Of these, 10 essential, 10 non-essential amino acids. Amino group itself clear that one amino group there and one acid group there. Among these, R group always varies one to another. Essential acids means they cannot prepare in our body. So non-essential means they can be prepared in our body. So no need to intake of these non-essential amino acids. All these components are attached to the central carbon atom is called alpha carbon atom. Based on the nature of the R group, a variety of amino acids classified. The R in the amino acid could be a hydrogen or aliphatic, aromatic or heterocyclic group. The amino group imparts the basic character to the amino acid. On other hand, carboxylic group gives the acidic character. Amino acids also referred as substituted methane. Each amino acid has free carboxyl group, a free amino group, and or as a distinctive side chain of. When we happen to see the glycine, as usual like methane, carboxylic group, amino group, and hydrogen ion, and in the place of R, H will be, that is glycine structure. When we come to the alanine, in the place of R, methyl group will be. That is the difference between the alanine and glycine. As we can observe, since its structure there is a small difference between the glycine and alanine, that is H and CH3. That's it. When we observe the valence, CH is attached to the alpha carbon and either side of the CH3 groups will be. Whenever we add the CH to alpha carbon and CH between, so it will become leucine. CH2 is the difference between valence and leucine. There is a small difference between the leucine and isoleucine. Just CH3 group addition of the CH, first CH of the this aliphatic group. That's all. That is the difference between the leucine and isoleucine. Whenever we compare the leucine and lysine, they are similar structure and similar formula will be, but the thing is the position of the CH groups will be difference. It is very easy to keep remember CH, CH2, CH3. That's all. So 1, 2, 3. That's all. In case of valence, CH, CH3, CH3 either sides, and in case of leucine, CH2 added between the alpha carbon and CH. And next, in case isoleucine, CH3 added to the after alpha carbon. That's it. In case of serine, we can observe the CH2 and OH group. Hydroxyl group will be added. On other hand, in the case of cysteine, CH2SS, here sulfur will be added. That is the difference between the serine and cysteine. Since these two are similar kind, we can keep remember easily. Serine. Serine means OH will be. So here cysteine means cysteine. There is a difference between the cysteine and OH is common by serine and threonine. Serine and threonine is hydrophilic and polar, means alcohol group will be. Hydroxyl group will be no. Threonine containing CH, CH3 and OH. Whenever OH is containing, that is a hydroxyl group, will be said to be alcoholic. Serine and threonine hydroxyl groups containing, whereas methionine and cysteine sulfur containing. Amino acid name itself saying that acids only. As there is a carboxyl group adding, this acidity also gradually improving. Whenever we check the structure of the aspartic acid and glutamic acid, there is a comparison, but only changes CH2 group will be added extra to the glutamic acid. In general, amino acids containing one carboxyl group, one amino group, then it will be neutral. So, as they are adding carboxyl group, acidity will be improved. More in number of carboxyl acids, more acidity will be. Like more amino groups, more basicity will be. Here we can observe that CH2COOH carboxyl group, whereas in glutamic acid CH2CH2COOH two groups. When we compare the asparagine and glutamine, there is a one change will be only CH2 group will be added to the glutamine. Having amino groups, these are basic nature and both are polar. Since adding the more amino groups in this asparagine basicity, improving in glutamine also. One extra CH2. When we draw the structure of lysine, we can find the four CH2 groups, after that amino group. So I'm addition of the amino group to the this lysine. It is a basic nature. In lysine 4 CH2 groups, 1 NH2, whereas 3 CH2 groups, 2 NH, 1 NH2. Now we have seen the arginine structure. When we examine the arginine structure, so we can find the 3 amino groups, 3 methyl groups. In lysine, 1 amino group, 4 methyl groups attached to the alpha carbon. This is a difference between lysine and arginine. When we see the phenylalanine, there is a one methyl group and one benzene ring will be. So that is aromatic. For this structure, if we add the OH group to the benzene ring, it becomes tyrosine. Tyrosine also aromatic. A small difference between phenylalanine and tyrosine, only hydroxyl group. Attachment of the hydroxyl group to the benzene ring, then it is called it as tyrosine. If we keep remembering OH group, that's it. Benzene ring attached to the alanine and non-polar. What do you think about tyrosine? Having OH group, that is considered as a polar, remaining non-polar, since it do not have OH groups. When come to the proline, so we can find the heterocyclic, means nitrogen and hydrocarbons both together will be in the one ring, so it considered as a heterocycle. Non-polar, since it consists only hydrocarbons and non-aromatic. In this amino group having the charged positively. Next to histidine, when we examine this histidine, removal of the one hydrogen from the alanine and attached the 5-membered heterocyclic group, lone pair amino group will be aromatic, and when positively charged left side amino group will have basic nature, and it is also positively charged. Left sided amino group will have 4 hydrogens. When we observe the tryptophan, so like histidine, CH2 group will be on chiral carbon, and also 5-membered heterocyclic link along with benzene ring. In one ring, hydrogen atom will be, so it is considered as a polar, rest non-polar, and also aromatic.